Okay. Welcome to the Public Health and Psychology 3 division. We have 4 presentations today. At the end of each presentation, we'll be taking questions from the judges and any of people from the audience who have questions. And before we begin the presentation, can the judges please briefly introduce themselves and mention their research field? So Morgan, we can start with you.
Yeah, my name is Morgan Snyder. I'm a second-year PhD student in the Department of Psychology and Neuroscience. My advisor is Dr. Gary Elkins in the Mind-Body Medicine Research Lab. So my main interests are mind-body medicine for different health outcomes like sleep and pain.
Thank you so much. And Kayla?
Hi everybody, I'm Kayla. I'm a third-year graduate student in the behavioral neuroscience track in psych and neuro, and I study diabetes and aging and how nutrition affects cognitive outcomes and affective outcomes in a preclinical model using mice.
Thank you so much. We really appreciate you guys coming to judge the presentations today and can't wait to hear your feedback. So our first presentation today is from Abhinav from Baylor University. And Abhinav, you can share your screen and begin whenever you're ready.
Thank you, Jasmine. So as Jasmine said, I'm a sophomore at Baylor University. And so I'll be presenting today on a project I completed during my time at Earl Hall as a freshman with the laparoscopy lab. And so the laparoscopy lab is a class for incoming freshmen, which allows them to develop proficiency with a laparoscopic simulator, a surgery simulator, as well as develop and participate in studies with that simulator. And so I completed this study as a freshman with my peers, Soumya Mahida Arvind, and our research mentor was Meredith Ullman. And so what is physician burnout? So here's some statistics that detail this concept. So 54% of doctors say they are burned out, 88% of doctors are moderately to severely stressed, and 59% of doctors wouldn't recommend a career in medicine to their children. And so all of these statistics detail a narrative that's becoming more and more prevalent in the field today, that doctors are overworked and that doctors are having significant mental health issues that are affecting their ability to operate properly. So physician burnout is one of a key cause to patient errors and errors within surgery, errors within diagnosis. And so we chose to explore roots of burnout within physician nutrition. And so our theory is that many of these mental health effects that are seen with burnout may have a dietary root in nutrition. So we attempted to find a suitable macromolecule that could help with on-duty snacking. And so here on this slide, on the left, we can see our laparoscopic simulator detailed. And so this is a simulator I was referring to earlier, in which undergraduate freshman students develop proficiency with. And so not only, so here in this box, we see the pegboard exercise, but students also complete exercises such as like cutting gauze and completing a suture with the laparoscopic Marylands. And so the Marylands are these long scissor-like bits. And at the insertion point between what we would call like the abdomen and like within the abdomen, we have these trocars here, which are basically the insertion device for the Marylands to enter the abdominal space. And so on the right here, we have the pegboard exercise in which students move the pegs from one side of the board to the other. And so and then they move them back. And so this exercise is taken from the fundamentals of laparoscopic surgery, which is a course for medical students, and really serves as the baseline for measuring dexterity and comfortability with the laparoscopic box. And so the proficiency with this exercise is scored based on time. So from the first touch of the peg to the last touch of the peg is how we score. So time starts at the first touch and stops at the last touch. And so that contributes to the exercise time, which you see on the bottom left corner. And then, so an error is counted as every time a peg drops outside of the field of vision. So outside of the field of vision and the Maryland, you're unable to pick up the peg, that's what counts as an error. And so each error contributes 10 seconds to the overall time, and the modified scores will be used to gauge proficiency. So detailed on this slide, we have the methodology for our experimental group, but first I'll go over our control group. So our control group consisted of laparoscopy students shown in the gray figures. And so these students, without any external factors, just came to the regular class and completed the pegboard exercise 9 times in rapid succession. And so and they were all scored identical to how they are scored in the experimental day. And so here for the experimental day, we had laparoscopic students. We had asked them to fast for 14 hours. And they subsequently filled out a pre-experiment survey, detailing did you eat, and confirming that they did fast, and some other factors were collected in the pre-experiment survey. And so then each of these students were randomly assigned to either a protein group or a carbohydrate group, as shown. And so depending on which group they got, they either received an isopure protein powder dissolved in water, or they received water with a crystallized sugar snack. And so each of these groups consumed their assigned macromolecule, and they went on to complete the pegboard 9 times in rapid succession. And not only were their proficiencies gauged, but we also recorded their energy levels throughout to see if, depending, because commonly we know that carbohydrate, digests faster, has a faster effect on energy levels. So we attempted to see that as well. And after they completed the exercises, they filled out a post-experiment survey, which asked about various dietary and lifestyle habits. For example, like whether they consume caffeine, whether they had fasted before, etc. So here we have 2 pieces of results that come from that post-experiment survey and correlated with our experimental times. So we found that that those who drank caffeine regularly, when fasted and with either protein or carbohydrate, on average found a higher improvement in time. So caffeine drinkers on experimental day had a significantly improved time. And so then for protein bars versus no protein bars, those who consume protein bars had a lower average time on experimental day than those who did not consume protein bars. And so this could have various effects with how these foods interact with the either carbohydrate or protein. For example, on protein bars, those who consume protein bars may have adapted to consuming, artificial, unnatural methods of consuming protein and that may have played a difference in how they digested the protein bars and how they did on the exercise. On this slide, we see our control versus experimental times. And so we saw on average that whether either if they can either carbohydrate or protein, regardless of which group they were in, we saw an increase in proficiency at times, so better times on experimental day. And so some objects of future work that could be completed, of course, is to repeat the study with more participants. I think our class was limited by the size of the laparoscopy lab and those who are proficient with the exercise. And we were also looking to monitor blood glucose with wearable technology. Since we're all undergraduates, we didn't necessarily have the permission to draw blood. So we were looking into techniques to use wearable technology, like wetless collection and monitoring of blood glucose. And so at that time, early 2019, there wasn't much. If there was technology, it would be very niche and very expensive to obtain. So I think since then, there's been many developments in wearable technology, and more companies have entered the space. And it seems like monitoring blood glucose will be possible without taking blood samples in the near future. For example, I think Apple filed for patent regarding this technology. And so another rectification that could have been made is to plan a study where control data is collected closer to the experimental day. Due to some of the bounds of our class, we had to wait 5 weeks after control day, 4 to 5 weeks after control day, to collect our experimental day. And so this just let in a whole host of factors in terms of students' personal life and other activities that would have been going on between control and experimental day that may have affected our results. And so planning a study closer where control data is collected closer to experimental day would be beneficial. And so I would like to acknowledge my research mentor, Meredith Allman, and our instructor, Dr. Marty Harville, and as well as Hallie Earl Hall for all the help they've contributed to the program at Baylor Laparoscopy. And just thank you to the entire Laparoscopy Lab family for allowing me to complete the study as a freshman. And yeah, so thank you for your attention, and I'd like to open the floor to questions.
Yes, thank you so much, Abhinav. That was really interesting. We'll start with, do the judges have any questions?
I do. Out of curiosity, for the fasting ramifications, were they allowed to drink whatever they wanted to drink during the fast?
No, they were not allowed to drink caffeine. They were allowed water, of course, but they were not allowed any substances other than water and celery.
Okay, cool. Thank you.
Yep.
I was wondering, could you just speak a little more to what they received as far as the snack goes on experimental day? So I know you said it was isopure protein, but could you speak a little more about the carbohydrate group?
So the carbohydrate group actually received pure sugar, pure crystallized sugar. And so, yeah, this is actually another topic for future work to blend and develop, a mixture of some of the carbohydrates. We felt that, if we did that, we would just be limiting and further dividing our subjects. And so in that, we wouldn't be able to, even if that was a good idea, we wouldn't be able to develop any significant data and the sample size would be too small. So.
Right. That makes sense. Thank you.
Thank you.
So would you guys consider using more complex carbs in the future that don't run through the system as quickly?
Yes, so that's the other aspect of the study. Since we had our class period, the 50 minutes to work with, we tried to choose we chose simple carbohydrates because of their quick quick effect on energy levels and quick impact. So in the future, we would like to explore complex carbohydrates and other macromolecules and their effect, and composition of fats, carbs, and proteins in a snack that may be beneficial towards doctors in long-term nutrition.
So I have just one more question. What would you say are the key takeaways from this study?
I'd say that we found some interesting data on how just a snack before operation affects affects performance. And so we didn't necessarily see much of a difference between carbohydrates and proteins, but we did see significance in that eating before and eating something before completing an exercise after a prolonged fast may make a difference. So
Definitely,
I'd say
definitely.
that's a key takeaway.
Yeah, definitely. Great job.
Thank you. All right, Jasmine, should I stop sharing?
Yes, go ahead. Thank you so much, Avinav, for the great presentation. Before we move on, judges, do you need any more time for your feedback forms? Good to go? Okay. So now we'll go to our second presentation, which is from Kara, Haley, and Lauren from Baylor University. You may go ahead and share your screen and begin when you're ready.
Can you guys see this? Yes. Okay, and then can you see my cursor as well? Okay, perfect. Okay, good morning. My name is Lauren Hogenacker, and with me are Kara Jones and Haley Turner. And our study looked at the effects of wearing a face mask on cardiopulmonary performance during exercise. So what prompted us to look into this study was the COVID-19 pandemic bringing about the need to wear a face mask to limit the spread of the SARS-CoV-2 virus. So as we know, wearing a face mask provides an extra layer over your mouth, and it's plausible to think that this may compromise breathing. So breathing, in short, is really just oxygen uptake, or VO2. So since it's possible that wearing a face mask may hinder VO2, our study really wanted to understand, does wearing a face mask really compromise exercise performance? So something important to note, during aerobic exercise, performance is measured as VO2 max. VO2 max is really the maximum amount of oxygen that's being inhaled. So this means if there is a reduced ability to uptake oxygen from a face mask, we believe VO2 may be compromised. And that's really why we used VO2 as one of our variables that we analyzed in this study. So it's also not known if there are long-term or short-term differences in how the body responds to exercising with a face mask. And due to time limitations, our study only analyzed the acute effects. So the purpose in completing the study was to explore the potential risks of wearing a face mask while exercising on acute cardiopulmonary measures and exercise performance.
Okay, so our study consisted of a total of 18 participants, 9 of which were male and 9 of which were female. The average age was 21.6 with a 2.5-year standard deviation. Our inclusion criteria consisted of college-age students, so ranging from ages 18 to 30 years, and they had to be able to perform sustained high-intensity exercise. Our exclusion criteria included anyone with orthopedic limitations to exercise, anyone who was pregnant at the time of the study, and anyone who was taking medication for any cardiopulmonary disease. So each participant participated in 2 trials that were randomly assigned either with a face mask or without a face mask. The face mask we used during the with face mask trials was just a standard disposable surgical mask, and there was a minimum of 48 hours between each of the 2 trials. So in order to test whether or not a face mask had any effects on exercise performance or cardiopulmonary measures, we had each participant complete 2 graded exercise tests. So in order to measure cardiopulmonary measures such as Vo2 Max, like what Lauren mentioned in the previous slides, and other measures such as respiratory rate and other things we'll talk about in the results section, we connected each of the participants to a metabolic cart, which can be seen in the bottom picture, and we had them run on a treadmill, which can be seen in the top picture alongside the metabolic cart. So throughout the exercise protocol, there was a steady increase in either speed or incline on the treadmill, and the participant completed the exercise until they reached fatigue. So once we obtain all of our data from each of the participants in the metabolic cart, we perform statistical analysis comparing the with face mask trial against the without face mask trials, and we used a 2-way repeated measures ANOVA using SPSS, which is a statistical software. So here are some figures that show how we connected the participant to the metabolic cart. So as you can see in both the without face mask trial, which is on the left, and the with face mask trial on the right, we connected each of the participants to this metabolic mask. And you can also see that there's a plastic tube that was connected to the end of it. So one side was connected to the mask, as you can see in the picture, and then the other end of the plastic tube was connected to the metabolic cart, which I showed in the previous slide. So yeah, and one thing that was really important during when we were fitting the mask was making sure that there was no leakage of air, because we want the air to stay within the tube and to go travel to the metabolic cart. So in order to control for this, we covered the end of the plastic part of the mask and had the participant breathe out to make sure that there was no leakage around the mask area. This was especially challenging for the with face mask trial. So we would use like tape and other measures to make sure that there was a perfect seal. So during both the with face mask and without face mask trials, we collected these following measures. So we collected heart rate using a polar heart rate monitor. We collected blood pressure using a manual cuff. And we collected blood oxygen saturation and perfusion index using a pulse oximeter. We collected Vo2 Max and the other cardiopulmonary measures we'll discuss later using the metabolic cart. And we also had each participant rate their perceived exertion using the RPE scale, which can be seen on the right, ranging from 6 to 20. So here is an outline of the exercise protocol. As you can see, there were 7 different stages, a resting period, and a warm-up period. So each of the stages lasted 3 minutes long. And during each stage, as it progressed through the stages, the speed increased. The incline also increased up until stage 3, where it leveled out, stopping at a 10% incline. And during each stage, we recorded heart rate, blood pressure, O2 saturation, perfusion index, VO2, and then RPE. The only exception was for blood pressure. As the participant reached fatigue, we stopped recording blood pressure because it was difficult as they were running. But yes, so each participant completed this protocol until fatigue, so they didn't always necessarily reach stage 7.
And so the results that we found after doing this graded exercise protocol is here, the notable variables. So with a face mask, VO2, which was the main indicator of aerobic performance that we were studying, we found was significantly lower, but that was only found at maximal intensity. In this chart here, denoted in red, and then also with an asterisk, are the statistically different values. So there is one value here under ventilation, but the most notable ones are over here, all at max between the with face mask and without face mask. So VO2, you can see here, statistically different. It was lower at max intensity with the face mask, so meaning that their aerobic performance wasn't as high as when they didn't have a face mask on. We found that additionally with the face mask, ventilation, which is denoted here, was significantly decreased by 24% at maximal intensity. And so ventilation is the volume that the participants are actually breathing into their lungs. So that was reduced at max intensity while wearing a face mask. And then RER, which is respiratory exchange ratio, it is the amount of carbon dioxide exhaled by a participant versus the amount of oxygen actually brought in and utilized by a participant. This was significantly higher in the without face mask trial. And so what that means is that they're reaching, when they're not wearing a face mask, they're actually able to reach a higher performance or more in that carb burning zone as opposed to that fat burning zone with the RER. And then respiratory rate in breaths per minute here, that was significantly higher when they were wearing a face mask. So they were breathing a higher number of times without wearing a face mask than when they were wearing the face mask. We broke it down by some graphs here, the things that I was talking about earlier. So respiratory rate in breaths per minute was significantly higher when they were not wearing a face mask. And again, that was only at maximal intensity. So you can see here, both plotted against each other without face mask and with the face mask. As the participants moved up in intensity and then breaking away here from 80% to them reaching the max, when they were not wearing a face mask, participants were seen to breathe more breaths per minute. For VO2, again, the good indicator of aerobic capacity was lower, and that was only found at maximal intensity, kind of similar to the graph that you just saw before. The two lines not statistically different here, and then once we reach above 80% to max, right here at this max intensity, when they were not wearing a face mask, participants were seen to be able to have a higher VO2, and so they were reaching a higher aerobic capacity there. Partial pressure of expired oxygen was significantly higher when participants were not wearing face mask at maximal intensity. So the partial pressure of expired oxygen is the participants are breathing out a certain amount of oxygen that's being picked up through the tube and into the metabolic cart. So relatively the same here, no statistical difference. And then at max, when participants are not wearing a face mask, they're seen to be breathing out more oxygen. So that means that they're not needing to use as much of that oxygen as opposed to when the participants are wearing a face mask, they're needing to use more oxygen, so they're not expiring as much as when they're not wearing the face mask. Going along with that, you kind of see the opposite of the curve we saw before. This is showing expired partial pressure of carbon dioxide. It was significantly higher when the participants were wearing a face mask at maximal intensity. And so here you see statistically different values here. When the participants are not wearing the face mask, they're expiring less carbon dioxide as opposed to when the participants are wearing the face mask. And then here, some of the other variables that we had mentioned that we looked at: heart rate, blood pressure, oxygen saturation, perfusion index, and then that rate of perceived exertion. We found no statistical difference between each of these with the face mask trial and the without face mask trial across the board during rest and then also max.
So after analyzing our results, we saw that the difference in VO2 was really only observed at maximal effort or above 80% VO2 max, which can lead us to believe there are unlikely to be detrimental effects of exercising in low to moderate intensity. Significantly reduced VO2 max with a face mask may be due to decreased ventilation. And the increased CO2 exhalation at maximal effort is believed to be due to excess CO2 being inhaled back in by the participant. And as Kara mentioned, heart rate, blood pressure, oxygen saturation, and perfusion index are unlikely to be affected by a face mask, as we saw with lack of significant differences between our two trials. So in conclusion, our study suggests there is no known concern for wearing a face mask at low to moderate intensity. And future studies are encouraged to analyze different populations, including people with pulmonary diseases, as well as analyzing different types of face masks and their effects. And that concludes our presentation. Thank you so much for your time and attention. Any questions are welcome.
Thank you so much. That was a wonderful presentation. I found that really interesting. We'll start with the judges. Do you guys have any questions?
I'm sorry if you already said this, but did you guys control for or ask what their level of exercise was habitually prior to joining your experiment?
So we didn't do any sort of surveys like that. We didn't include in here because we didn't actually run any statistical analysis on it or actually really take it into consideration, but we did do DEXA scans on them. And so that just kind of looked at like body fat percentage, muscle that they had, bone mineral density. They were all relatively active participants according to that, but no, yeah, we didn't ask any if they did participate in any sort of activity prior.
Okay. Thank you. You guys did great, by the way.
Thank you.
Yeah, great job. Did you have all participants wear the same face mask during your study, or did you let them wear the one that they wear um, like in daily life?
Yeah, so we had all the participants wear the same surgical disposable mask during the with face mask trial.
Awesome.
We provided it for them. And so like
Okay.
they discarded it at the end.
Perfect. Do you think there's any limitations to your study?
There's definitely some limitations. I think that so the original idea that I had was for kind of doing. So I teach F45 and the slick, and so I wanted to kind of look at if they were wearing the face mask and they're doing this high intensity exercise. The limitations I think with the treadmill is that they were starting at like rest seated, and then it slowly built up. So maybe translating that to different actual real life situations where people are actually going to be completing, most people don't complete a graded exercise test, but that was what we studied. So maybe in that sense, more translatable to real life.
Right, right. And I was also wondering, because the picture you had of the equipment that you use to measure Vo2 max, that includes a mask as well. So I was just wondering like how generalizable that is to real exercise settings. But I think it's really interesting that, for example, you didn't find any differences in ratings of perceived exertion either. So they weren't even perceiving it to be harder. So very interesting.
Mm-hmm. I will say anecdotally, we would ask the participants how they felt after, and oftentimes they said that they felt like they could do more, like their legs could do more, but they just like couldn't breathe at a point. And a lot of participants would sweat through the mask, and that made it also really difficult. And then by the time they took it off, they were just like, thank goodness, fresh air. So
Yeah, because it looked like kind of an uncomfortable situation.
yeah.
So,
One
yeah.
of the participants described the feeling as being waterboarded from the sweaty
Wow.
mask.
Wow, that's
He was
crazy.
fine. We made sure he was fine. But
Oh yeah, for
It's
sure.
okay, but
yeah, he's good.
I would definitely say that was a limitation too. Like even like without the face mask, the metabolic mask can be kind of uncomfortable in itself. So I mean, most participants kind of expressed that, as they started going, as the protocol progressed, that they started to get more comfortable with it. But yeah, I'm kind of curious to know, like, had that not been a distraction, if they could have maybe gone longer.
Right, right.
Do you guys think you would see similar results for a strength workout versus the cardiovascular workout?
I think that'd be interesting. I'm not sure what we would see from it. Because definitely, so VO2, I'm sure you would have to use a different parameter. I don't know what you would use, but that's for aerobic performance. And so a lot of those variables only translate to when you're running, as opposed to you do a very different, like physiological, big physiological differences between strength training as opposed to running. But that would be interesting to look into for sure.
Awesome. Well, thank you so much, Kara, Haley, and Lauren. That was a fascinating presentation. Judges, are we ready to go for the next one? Awesome. So we'll start the third presentation now. Our third presenter is Paige from Baylor University. You may share your screen and begin whenever you're ready.
All right, is everyone able to see this? Awesome. Well, hello, my name is Paige Freeberg. I'm a junior psychology major with a dance minor at Baylor, and I'm going to be presenting over the impact of stress mindset on cardiovascular and psychological habituation to a repeated stressor. And this is a poster that I presented, not with a presentation, but just viewed at the American Psychosomatic Society conference in December. So this is just a broken down version of that poster with kind of my commentary and more of a presentation style. And I'm a member of the Baylor Behavioral Medicine Laboratory, so I'd like to give special thanks to Dr. Ginty and Dr. Young, and then Dr. Williams at the University of Birmingham, as well as Alex Tyra for all of their help with this. And I wasn't present for the data collection, obviously, because it was before I was there, but yes, so I'm excited to share this with you all today. All right, so do you think stress is something that's good or bad? So stress mindset involves viewing stress as enhancing or debilitating. This kind of work emerged from Dr. Crum at Stanford, and it kind of emerged from the kind of notion that stress is viewed in a pretty negative light in society. It's usually something that people want to avoid. They say it's something that's bad, that we don't want to have anything to do with. And so when you think about a mindset, that's a way of mental framework or a mental categorization of something. And there's mindsets for a lot of things, like expectations for treatment outcomes, expectations for what you think certain things will do. And the same thing applies to the way that you think about stress. And it's different than stress coping and stress appraisal, which are often used in kind of this behavioral medicine or stress reactivity hypotheses. So stress mindset refers to these attributes and expectations you have about stress, regardless of whether or not you're stressed right now. So I may not have a test coming up right now, but I can still say whether or not I think stress is good or bad. And so stress mindset will kind of inform the coping strategies that you use. So it's very different than coping. It's more of a general thing that you think about stress and whether you think it's good or not. And it's also different than an appraisal of a stressor. An appraisal is kind of a specific attribution that is very stressor specific. So you might appraise a certain stressor as really stressful, but then be like, I think stress is something that's good and can help me. And then that can actually impact the way that you're reacting to that stressor and vice versa. So stress mindset is definitely different than coping, different than appraisal. It's kind of a way of evaluating the nature of stress itself. And so research in the past has experimentally manipulated stress mindset. So they've kind of presented whether it's a video or kind of a prompt where people read facts about stress and they're like, stress is something that's positive and can help you, or stress is something negative and can hurt you. And so when research has done that, they found that it is related to physiological and psychological responses at one time point for a stressor. But research has not looked at responses to multiple stressors. And research by Hughes et al. Is talking about habituation and how that's important to look at two different stressors. And so habituation is basically looking at how you're adapting to multiple stressors. So a good adaptive stress response would be that there's reduced cardiovascular reactivity and reduced perceptions of stress after a second stressor. And I think that looking at habituation is something that's really interesting because often in stress research, if you only look at it at one time point, it can look like someone's like excessive or hyperreactive, but if you look at them at a second stressor, their stress levels actually go down. So that kind of research looks at a lot of different patterns across multiple stressors and how oftentimes one stressor doesn't really give us the full picture of how someone responds to stress. And then there's research that's talking about kind of combining a specific appraisal of a stressor and stress mindset to kind of optimize stress. So looking at how we can use stress as a way to help us and a way to use those resources in a way that can, because sometimes stress is inevitable, it's unavoidable, and how we can use these resources in a way that we respond and think about stress to actually have not only a more adaptive response to stress physiologically, but then psychologically have all the resources we can to help us utilize stress in a positive way. And so that's why it's important to be looking at not only stress over 2 time points, that habituation, which is definitely relevant to cardiovascular disease pathways, but also look at stress optimization, which is kind of the more psychological side of everything. All right, so based on all of that, the goal of this was essentially to examine whether stress mindset is related to habituation in both physiological responses, so cardiovascular reactivity, and then perceived physiological responses, so how you perceive your physiological arousal and whether you think that's helpful or hurtful, and then your perceived stress across 2 identical acute psychological stressors. And instead of manipulating stress mindset experimentally, we're looking at it dispositionally. So we gave a questionnaire about stress mindset, and it's kind of everyone's just natural ways of thinking about stress. We didn't manipulate it, which is different than prior research. So really looking at it in a dispositional way and then looking at it across 2 stressors is something that's really unique about this study. All right, so we had 446 participants in the study, 62% female, 66.1% Caucasian, and 17.3% Hispanic. They completed these 2 identical stress tasks, and the stress task they did was the PSAT. So it's a standard mental arithmetic task that people tend to rate as pretty stressful, and it contains elements of self-evaluation, social evaluation, and time pressure to really elicit a stress reaction. And so as you can see in this figure here, They had a 10-minute baseline where blood pressure was measured. Then they went into that 4-minute Passat, and then they had a rest and repeated that first protocol. So they had the baseline and did the second stress task. And heart rate was recorded throughout. And then the reactivity was calculated separately for each task. So they took the stress levels minus the baseline levels of heart rate, those averages. And then before and after each stress task, these perceived physiological arousal intensity, interpretation, and then stress intensity and interpretation were evaluated. And this study is really only looking at the post-stress task, so looking at these 2 time points after they experienced the stressors. And so the intensity was just essentially how stressed did you feel? How stressed are you right now during this after technically you did it? The interpretation was whether or not you thought that was positive and whether or not. So if I say I'm like, yeah, I'm really stressed right now, and then the question is, did you think that helps you perform? That would be the interpretation. So saying how facilitative or debilitative you thought stress was during the stress task. All right, and so this table is showing the main effect of time on all of the outcome variables. And so as you can see, there is adaptive habituation and heart rate reactivity, perceived stress, and physiological arousal intensity. So there is a significant difference or decrease technically in these outcomes because it was adaptive between time 1 and time 2. And there is not a main effective time on perceived physiological arousal interpretation or perceived stress interpretation. So when we're not looking at stress mindset, we're seeing a main effective time on this heart rate reactivity and the intensity of people's physiological arousal and their stress. But we're not seeing habituation over time or changes that are significant between their interpretation of stress and physiological arousal. So whether or not they think that's good or bad, which is different than stress mindset. It's just one of the questionnaire facts that are given during it. And all this right here is just essentially saying that stress mindset was not related to these variables. So stress mindset was not related to heart rate reactivity, the perceived physiological arousal intensity, or perceived physiological arousal interpretation. So this is essentially saying there's no effective stress mindset on habituation of these variables. And these are the physiological variables and perceived physiological variables, which is interesting because you would think that the way you think about stress in general would affect the way you react to these specific stressors physiologically or how you perceive that physiology. But this was finding that there is not a relationship there. And so the next one, there was a relationship between these psychological perceptions of stress and the experience of stress. So for figure 2, we have on the x-axis stress mindset. So a higher score indicates a more stress is enhancing mindset. And the y-axis is kind of the habituation and stress intensity. So time 1 minus time 2. So if people viewed stress as less intense at the second time, there's going to be more habituation. And so there was habituation in stress intensity over time. As we saw, there was a mean effective time on that. And there was also a interaction between stress mindset and time for this perceived stress intensity. So on the left are figure 2. So this is saying that stress mindset impacted habituation of perceived stress intensity. Basically, people who had a more stress is enhancing mindset perceived stress to be as less intense after that second stressor. And so on the right for figure 3, the y-axis, we have perceived stress interpretation habituation. And same thing as before, a higher score would indicate that more habituation, but we didn't have a main effective time on perceived stress interpretation. The main effects were only on the heart rate reactivity and the actual stress intensity and physiological arousal. So even though there was not a general pattern of perceived stress interpretation that was actually demonstrating habituation in the sample in general, so it didn't interpret stress to be more facilitative at the second time. Stress mindset did influence this habituation and stress interpretation. So people had a more stress enhancing mindset, interpreted their stress as more facilitative after the second stressor, even if the sample didn't overall across time, which is really interesting. So in conclusion, stress mindset was not related to habituation of heart rate reactivity or perceived physiological arousal intensity or interpretation over 2 identical stressors. But stress mindset did impact psychological habituation. So it did impact the perceived stress intensity and perceived stress interpretation. So participants who viewed stress as more enhancing rated that second stressor as less stressful and interpreted that stress as more facilitative the second time. And that stress is enhancing mindset that questionnaire, it's really an 8-item questionnaire that's asking, I think stress is something that's good and benefits my learning and growth, or I think stress is something that's negative and should be avoided. So that stress is enhancing mindset. So people who scored higher on that stress mindset questionnaire, they were viewing stress as more enhanced. They were A, viewing stress as more enhancing, and B, rating that stress as more, so they viewed it as more, they view stress in general as more enhancing. They rated the second stressor as less stressful and interpret that stress as more facilitative. And so this is basically saying that general views of stress do impact habituation of perceived stress, but not of perceived physiological arousal or heart rate reactivity. And it's interesting because this stress mindset did not impact actual physiological reactivity or perceived physiological arousal, but it did impact psychological perceptions of stress. So one reason for this may be in previous research, they were kind of looking at responses like cortisol, or they were looking at anabolic growth hormones, which are more having to do with the HPA axis and not so much of the sympathetic nervous system reaction, which is the sympathetic nervous system reaction in stress, as we know, is something that's much faster. So it may be that the stress mindset is not influencing that specific physiology. And also, another reason may be that people who have a more stress enhancing mindset in general have better psychological adaptation to multiple stressors because they have a better view overall, and it influences then a more positive reappraisal of a specific stressor. So a strength of this was we had a large sample size, 444 participants, and using those two stressors. Then a limitation was that this is primarily college students, and that there was a lack of diversity in the sample. Diversity in research samples is something that always can be improved upon. And when there's diversity in research samples, in researchers, in perspectives in research, it really is strength. The research itself is strengthened. The inclusivity and its application is so important. So that's something that can definitely be improved on. And then we didn't control for other factors such as their mental health, their neuroticism, and for like social desirability and everything like that. So the final takeaway is stress mindset was not related to habituation of heart rate reactivity, perceived physiological arousal intensity, or perceived physiological arousal interpretation, but it did impact habituation of perceived stress intensity and perceived stress interpretation. And just kind of quick future directions. This is kind of emphasizing the importance of examining multiple stressors, not just using one stress time point. And then also the way these psychological factors, such as stress mindset and the way you're viewing stress, influence your habituation, both physiologically and psychologically. And future research should examine pathways. So why does stress mindset not influence physiology, but it does influence psychology? So thank you so much.
Thank you so much, Paige. That was really interesting. I loved hearing about that. We'll start with questions from our judges.
Great job. That was a lot of information, but you handled it really well, and I liked how well you walked us through all of that. I've done some preclinical stress models, so I'm not super big in this area. This may or may not be a silly question then, but did you guys or did your lab ask or control for physiological exercise and how it might have correlated with their results and or psychological perceptions of stress? Just in me asking that is because we did some stuff with mice where we looked at how exercise or habitual cardiovascular exercise helped buffer their responses to stress at a more physiological level or neurological level, really. But so that's why I'm asking.
No, that's a really great question and a good point. And even as I was watching the last one, we used the same exact like metabolic card for the VO2 and everything. And that wasn't included in this study, but that's why watching the last one, I was like, oh, I'm familiar with that. That's so cool with like the RER and everything. And I'm pretty sure that we don't ask them how regularly they exercise, which I agree would make a difference, I would think, in the way people respond to stress, because it has to do with like the health of their heart and their whole cardiovascular system and their fitness. And so I think they get the VO2 and everything, but in general ones like this, I don't think people are asking how often they exercise, and kind of controlling for how they view stress in general was the stress mindset. I'm pretty sure that's the only thing, because the stress mindset questionnaire was kind of asked at the end. And so they, not the end of the study, end of their kind of, after they did everything, it was like the trait questionnaire part. And so they asked that stress mindset questionnaire there. But that was kind of the only thing to control for how they view stress in general, because otherwise, normally when people are, I guess, are asking about stress, it's at specific stressors. So all those questionnaires where they're asking their intensity and interpretation, that was asking in relation to after the stressor, after the BSAT they had just done. And so that was kind of what was interesting is, I guess the stress mindset was the thing that was controlling for how they view stress in general, and it wasn't impacting their physiology or how they perceive their physiology, but it was impacting how intense they view stress and how they interpret it. Did that answer your question? That's my best understanding.
Yeah, no, that was great. Thank you.
Do we have time for another question, Jasmine? Is that okay?
Yes, we can do one quick one. Go for it.
Okay, I'll just ask a quick one. Can you talk a little bit more in detail about the PSAT, so the stress test that you used in the study? And I'm just curious if you believe that the PSAT is a good like lab, if it's a good generalizable application of stress.
That's a great question. So I've never personally ran the PSAT. I've only ever done like Stroop tests and like IMSIT where they're on the computer and doing it, which I think that is like what you mentioned with using these lab stressors, like are they generalizable? Because I do know, for example, the PSAT, that one I've taken it before and I can say it's very stressful when I'm doing it. You know, you're counting backwards and you're having something buzzing you at random times. You have there's a social evaluation, you're being compared to other people. So, and you have to stare at a mirror for the whole time, look at your, like, it's definitely, they definitely throw in all the elements to make it as stressful as possible, I feel like. So definitely in previous studies and research has elicited stress responses from people. So I know that's pretty supported. But as far as the generalizability to stressors outside the lab, it's like, I feel like that's the question I always ask is, you know, in labs, we have these standardized protocols, which is great. And like, that's the benefit of doing all this in a lab is you get all these controlled environments, but then how much does that generalize to these daily and more importantly, like chronic stressors that people experience all the time? Like you're not necessarily taking a facade every single day, but people are, you know, stuck in traffic. They're having interpersonal conflict every day. So I do think the more clinical application of that is so important. And I don't think it's there yet because how can you generalize something that's so different from what real life is like when it's in such a controlled environment? So that was a good question. Did that answer it?
Yeah, it does. Great job. Thank you.
Thank you.
Well, thank you so much, Paige. We had a bunch of wonderful presentations today. I'm going to go ahead and end the recording really quick.